Good morning to everybody. I'm Berідзе, the pupil of the MAC Young Researcher, KPND Odessa Ecological and Naturalistic Center Felina. I want to present to you my scientific research. The study of the influence of coniferous plant extracts on the germination of wet and reddish seeds. Physiologically active substance of coniferous plants that grow on the territory of Ukraine in park, forest strips, and forest massifs can be used to create drugs with stimulating and herbicidal action that would be safe for humans and the environment. Traditional herbicides can also cause various diseases, including cancer. In world practice, a biologically active drug from extracts of some coniferous plants is successfully used. For example, the successful Japanese drug HB 101, but these are individual drugs and there are no domestic ones at all. Therefore, the aim of our work was to study the action of physiologically active substance of extracts of blueberry and virgin juniper, Himalayan pine, black pine, cedar of Lebanon, and cedar of Atlas in different concentrations and under different conditions to create physiologically active drugs. The task of our research was analysis of scientific sources by research topic, conducting a series of spring, summer, autumn, and winter experiments to investigate the influence of coniferous extracts on the growth and development of reddish varieties, national and Saxa, and soft wet varieties, Arnautka and Luzanska. Research had an application of coniferous plants extracts in vivo, analysis of the obtained data, and their use for the development of drugs with herbicidal and stimulating actions. The objects of the research were berry yew, virgin juniper, Himalayan pine, black pine, cedar of Lebanon, and cedar of Atlas plants. We also use soft wets of the Arnautka and Luzanivska varieties, radishes of the Saxon national varieties as an object of research. The work was carried out in UNESCO Park Monument of Garden and Park of Art of Regional Humanitarian Center and the Odessa Ecological and Naturalistic Center Felina during 2022-2024. In 2023, we used the seed of two species, wet of the variety Luzanivska and reddish of the variety Sexa, in studies of the action of the extract of coniferous plants. From the beginning, we soaked the seeds in 3% hydrogen peroxide for 15-20 minutes. Then 10 seeds were placed in petri dishes. Before that, we made extract from the leaves of the Himalayan pine in different concentrations and other coniferous plants in different concentrations. We ground the leaves in a mortar and mixed them in the concentrations indicated above at a water temperature of 80-100 degrees Celsius. Work on the study of the effects of coniferous plant extracts began in March 2022. For 2 years, we studied a lot of literature and performed more than 200 experiments. A number of factors influence the effect of physiologically active substance of conifers on wet and reddish seeds. One of these factors is the use of leaves of different ages. In spring experiments, we used old leaves, and in summer experiments, we used young leaves of berry and Himalayan pine plants, but only a different stage of development. The second factor that influenced the results of the experiments was seasonal fluctuation in the context of physiologically active substances like terpenes and other. The third factor, or rather a set of abiotic factors, such as insulation, precipitation, and temperature. The fourth factor that influenced the different results of the experiments was different concentrations of the extracts. Small concentrations such as 10 gram per 250 milliliters and 1 to 10 showed either a weak inhibitory effect of or were equal to the control, and in many cases stimulated the growth of plants compared to the control. An interesting physiologically active effect was observed with extracts of cedar of Lebanon and Atlas in concentration of 10 grams of leaves per 250 milliliters of water. In all experiments, we recorded a strong stimulation of seed germination on the extracts of cedar of Lebanon and Atlas cedar. Experiments on the use of black pine and Lebanon cedar leaves in vivo showed that the dilution of 50 grams of leaves, 25% black pine and 25% Lebanon cedar, per 10 liters of water was infusion from 2 to 24 hours stimulated the growth of young shoots of Himalayan and Atlas cedar. Thorny spruce leaf extracts were used for root and further fertilization. The greatest effect was obtained when we used in combination with the mineral fertilizer master for conifers by the Villarga company. Yuberry extracts in concentrations of 20-30 gram per 100 milliliter in June-July can be used to create herbicidal preparation. Compared to the control, Yuberry extract inhibited the roots of Saxa radish by 600%. This was confirmed by many experiments in which Yuberry completely inhibited the growth of the roots of the Saxa radish. Also in experiments comparing Yuberry extracts and Roundup herbicidal drug, Yuberry showed the same effectiveness as the herbicidal drug. Himalayan pine extract in concentration of 10 gram per 250 milliliter and in a dilution of 1 to 10 stimulated the growth of the roots of wet seeds of the Luzanivska variety and radish of the Saxa variety in June and August. The extracts of cedar of Lebanon and cedar of Atlas stimulated the growth of roots of Luzanivska wet in August, October, November, and December. Compared to the control, the extracts of cedar of Lebanon stimulated the growth of wet roots in August by 667%. Compared to the control, the Atlas cedar extract stimulated the growth of wet roots in October, November by 213%. Thank you for your attention.